Hello, my name is Yinji, and today I will be discussing RNA. So, before we dive in straight into RNA, we kind of need to have a little bit of overview over DNA and the other processes that I will be talking about. So, DNA encodes genes for traits by making RNA through a process called transcription, which I'll be talking about later in this video, and then a process called translation takes place from RNA to make protein, and I'll also be discussing that in a few moments. But first, before we dive into RNA, a little bit more information about DNA. DNA stands for deoxyribonucleic acid, and it is a hereditary material responsible for making up humans, animals, and all living things. And everyone has unique DNA, and that's what makes us different. So, DNA determines how someone looks, behaves, and functions. It is also responsible for causing the genetic differences uh, for diseases such as cystic fibrosis and Down syndrome. Oh, and this is possible through genes, which are little sections of DNA that code specific characteristics. So, as I mentioned earlier, DNA is hereditary, so DNA is passed down from generation to generation, and genes from the mother and the father are combined to form genes for their offspring, half mother and half from the father. So DNA is kind of known for having this distinct double helix pattern. It's also referred to as looking like a ladder that is crossed. And DNA is made up of four different bases: adenine, thymine, which pair together, and cytosine and guanine, which pair together. And they're paired through hydrogen bonds. Okay, so now we're going to transition to talk about RNA. A little bit of historical context first, though. RNA was the first genetic molecule to arise about 4 billion years ago. It started out as simple and unrefined and eventually evolved to be complicated and compound. And this is what a structure of RNA looks like, also known as ribonucleic acid, and I'll be going more in depth about RNA. So, a couple of differences between DNA and RNA, first and foremost, is DNA, as I mentioned earlier, is double stranded, RNA is instead single stranded. Um, RNA is also made from ribose sugar, and which makes it more unstable than DNA, whereas DNA uses deoxyribose sugar, deoxy literally meaning removal of oxygen. And another big difference is RNA, instead of having thymine as a base, has uracil as a base. Thymine in DNA is replaced by uracil. So the four base pairs in RNA are adenine, uracil, cytosine, and guanine. Um, Cytosine and guanine still paired together, but adenine is paired with uracil. There are three types of RNA also. We have messenger RNA, more commonly known as mRNA, transfer RNA, known as tRNA, and ribosomal rRNA. So mRNA transfers code to ribosomes for translation. tRNA brings anticodons that carries amino acids into the ribosome for protein synthesis. And rRNA makes up the ribosome, which is where the name comes from. So, in transcription, essentially the DNA strand unzips and acts as a template to make an mRNA messenger RNA sequence, which is used in a process called translation. And to briefly explain translation, when mRNA goes to the ribosome, tRNA comes into the transfer RNA, excuse me. Then anticodons attached to tRNA connects to the codons to form chains of amino acids, which become a polypeptide known as a protein, which is able to carry out the necessary functions for a human body or other living things. And here's an example of a DNA sequence to mRNA sequence, a translation. As we can see in the DNA, we have the four base pairs: A, C, G, T, adenine, cytosine, guanine, and thymine. And then in mRNA, we have uracil. Oh, we have uracil instead of the thymine. And then we can see the pairing as well. So adenine with uracil, cytosine with guanine, guanine with cytosine, and thymine with adenine. Next, I'll be talking about mutations. Mutations, what are they? Mutations are changes in DNA code that occur during DNA replication. How can they be corrected? Through proofreading and mismatch repair. DNA polymerase removes incorrectly added nucleotides. And mismatch repair repairs insertion, mismatch, and deletion of DNA bases. And how do they manifest? They manifest through cancers and other genetic disorders. And here are the different types of mutations. Most of them are pretty self-explanatory, but I'll also go over them. Deletion, part of the chromosome is deleted. Duplication, part of the chromosome is duplicated. Inversion, chromosome twists and inverts code. Translocation, traded between chromosomes. And that is the end of RNA. Thank you for watching.